All right, so in this lecture, we're going to be doing a couple different experiments to basically understand some of the transport mechanisms that occur within the cell. Okay, and so the first experiment that I have for you is what I have here, this beaker, and you can see these two different layers here within the beaker. Okay, so let me tell you how all this happened. So the way I went about doing this. So the first thing, I added a little bit of olive oil here to the beaker, and then I added some water. So I added the water, mixed it up, and it separated into these two components. Okay. So why is it that it's separated into two different layers and it's not what we would call a homogeneous mixture? Why is that the case? Anyone know? It's based off of polarity. Okay, so water is a polar molecule. Okay, and as you can see on the board, you can see the partial negative and the partial positive. Right, so the electrons are inducing what we call a dipole moment. So water is a polar molecule, and the olive oil, it's a lipid. And so lipids are nonpolar. So the entire purpose of this is to show you that the function of the plasma membrane for the cell, right? Because the plasma membrane, it's functioning to separate the components, the inside and the outside of the cell. So when we say it's inside, we say it's intracellular. On the outside of the cell, that's extracellular. Make sense? Okay. So the next question that I have for you is, why is it that the water is at the bottom and not at the top? Because remember, I had the oil in there, the olive oil, I added that first. But then when I mixed it, it separated. Why is the water on the bottom? Exactly, it's for density, right? Whatever's more dense, that's what's going to sink to the bottom compared to whatever's at the top, that's going to be less dense. Okay, so does that make sense as far as the separation and how this relates to the plasma membrane? Yes? Okay, let's move on. All right, so in this next experiment, what I have here, so this is my beaker of water, and this is my tea bag here. Okay, so let's do a couple basic chemistry terms. So you have a solute, you have a solvent, and you have a solution. So when I put this tea bag here into this beaker, what do I consider, what is this called? The solute. And what is the water? Solvent. When they're added together, we form a solution. Okay, so we understand the basics here. But the two concepts that I want to explain with this are diffusion and osmosis. So I've already done this experiment, and what I have here, this is a scale, obviously. And I want to weigh, what I want to show you is the difference in weight between the dry tea bag versus the ones that are already wet, that I've already dunked in here. Okay, so if we weigh this, the scale shows 8 grams. Okay, now if I take it off and I place the wet tea bag here, it's 31 grams. Wow, so that's a huge difference, right, when it's dry versus when it's wet. And so why is it the case that this tea bag weighs so much more compared to the dry one? Because it soaks up all the water through what type of process? Osmosis. So what this is representing here, what would we compare this to? The plasma membrane, right? So when I add when I add my solute, so let's go ahead and do this. When I add my solute here into the beaker, so I'm going to do this for a little bit. Is the water going to change colors, or is it going to be the same? It's going to change colors. And so why does the water change colors? Because of diffusion. So we've just learned two processes that are happening here. So osmosis is happening because we're adding our solute. So the water enters into the tea bag via osmosis. And diffusion is happening because what we call this, what's inside the tea bag, is that a low concentration or a high concentration? It's a high concentration. So it's going from a high concentration to a low concentration. And so you can already see the color is starting to change because it's diffusing out, right? But at the same time, it's still separated. None of the components within the tea bag are actually getting out because it's separated by the actual bag itself, or our plasma membrane per se. Okay? So any questions about this? This makes sense? Yeah? All right, so let's move onwards. So now what I have here, this is a hot beaker filled with water, and this is a cold beaker filled with water. And so what I'm demonstrating here, this is diffusion. Okay, but we're adding a heat source. And so this is why the experiment, you have this set up on your table. And so let me explain to you what's going to happen. So I want one person to get the red dye and one person to get the green dye, or whatever color it is that you have on your lab table. So go ahead and do that now. So someone grab that other one. Yes. Okay, so let me explain what we're going to do. So we're going to add a drop of each. So one will be in the hot beaker and one will be in the cold beaker. Okay. So I want you to do this at the same time, but not yet. I know you're excited, yeah. So the reason that we're doing this is to understand kinetic energy and how this can increase the rate that we reach equilibrium. So let's talk about a prediction. So when we add a drop of one drop into the hot, one drop into the cold, my question for you is, which one is going to diffuse at a faster rate, the hot one or the cold one? The hot one. Why is that the case?
It's increased kinetic energy because of the heat.
Exactly. So that increase in heat, it's causing these molecules to bump into each other, right? They're traveling at faster rates. And so the rate at which we reach equilibrium, the process of diffusion, it's going to happen at a lot faster rate. Okay? So my next question for you is, so then what's the difference between osmosis and the simple diffusion? So with osmosis, we have a semi-permeable membrane. Okay? But just for simple diffusion, which is what we're doing now, there's not a permeable membrane. It's just diffusing right away. Okay, so that's just another thing that I wanted to point out. Okay? So, are you ready? Yeah? Okay. So go ahead, put a drop in of each. All right, so Were our predictions correct?
Yes.
Yes, they were correct. Okay, so now let's move on to our next experiment. So the next process that we're going to discuss is what's known as filtration. Okay, and so how this rate relates to biology, we're going to discuss the kidney a little bit. Okay, so this here is my kidney model. All right, so just some of the basic anatomy. This is the cortex and this is the medulla within the kidney. Okay, fairly simple here. And this model that I have here, this is essentially a blown up portion, what it looks like here. So this part is the cortex, what I was just showing, and this part is the medulla. All right, so we're not learning, everything about the kidney and all that stuff yet, but it's just to get you to understand what it is that I'm trying to communicate as far as filtration. All right, so if you look here at this model, you see these little capillaries here, these little red capillaries. Well, in order for urine to form, it has to get into this part, which is known as the collecting duct. You don't need to memorize that or anything, so don't freak out about it, okay? But the urine has to get here. And so when you go pee, right, there's no red blood cells in your urine, okay? But the reason that there's no red blood cells in your urine is because we have what's known as this filtration membrane here. So it's preventing the red blood cells from getting into here. It's just blood that's circulating, and then it's going to get back into the venous system. Make sense? Yeah? Okay, so we're going to compare this with our simple little apparatus here, our experiment here. So the filter paper, what we have here at the top, that's what would represent like the filtration membrane of the kidney. Right? It's preventing the red blood cells from getting into the collecting ducts. Okay? And then what you can see here on the inside, the components that were in the tea bag, that's here in the top. So that we could represent that as the red blood cells. Okay. So what we're going to do in this experiment, we're going to add the water here onto the top. And what is changing is what's known as the hydrostatic pressure. Okay, so let me explain the hydrostatic pressure. So what's causing the fluid to move down, obviously we have that filtration membrane preventing the stuff that's on the inside from getting in, right? But what we're doing, what's happening is this potential energy of all this water that's here, it's being converted into kinetic energy once it starts to drop. Okay, because gravity is what's helping to bring this down. Make sense? Yeah? Okay. So let's go ahead and start our experiment. So what I want you to do, you're just going to get this beaker here with water and you're going to pour it at the top. And do you think is the water going to change color?
Yeah.
Yeah, it's going to change color, right? Because we're still getting the components that are in here, but it's just not the actual what's in the actual tea bag itself, right? Because we have the filtration membrane. So let's go ahead and start the experiment. So just go ahead and add it, and you'll see once you start to fill it to the top, right, all that potential energy is being converted into kinetic energy. Let me turn this on. So we'll just let it slowly drip here. Are there any questions for me? No. Okay. All right. So that's it for this lecture.